Welcome to the Health Headliner podcast by Tudu Digital. Our content is informational only and not medical advice. Please consult your provider. Now, let's dive into today's headlines.
Have you ever had one of those moments where something just clicks in your mind, like a mental snapshot?
Oh, absolutely. While other experiences just kind of blend together, right?
Exactly. It's almost like our brains instinctively know when to hit save on a particular moment.
Yeah.
So what if those save points are actually how our brain, organizes our entire life story?
That's a fascinating way to put it.
Well, today we're diving into a really groundbreaking discovery that's changing how we understand human memory works.
It's quite exciting stuff.
It really is. For the very first time, and this is key, in a natural sort of real-world setting, researchers have identified a key neural mechanism, basically how our brains segment and store experiences.
Right. It's not just about passively recording.
No, think of it more like an orchestra. Your brain is conducting and organizing everything into coherent, lasting memories.
That's a great analogy.
We've pulled insights from a health news article, and importantly, we've gone deep into the actual scientific paper in Nature Communications that details this whole experiment.
So we've got the details.
We do. So let's unpack how this memory orchestra actually performs its magic.
Okay, so the core mission here, the real challenge they tackled, was understanding how we go from this continuous stream of life
Yeah, just one thing after another.
to these distinct episodic memories we can actually recall. Specific events, moments. How does the brain do that segmentation?
And this wasn't like completely out of nowhere, right? There was groundwork already laid, especially from animal studies.
Definitely. Previous research, mostly in mice, strongly suggested that these things called ripple-type brain waves,
Which are?
they're basically just really rapid bursts of high-frequency electrical activity in the brain.
Gotcha.
And these waves seemed crucial for memory formation. They appeared to coordinate communication between the hippocampus.
Brain's memory hub.
Exactly. And other areas like the temporal cortex and frontal cortex. They help kind of integrate new information and strengthen connections, make memory stick.
But studying these ripples in humans, that's been the tricky part.
Hugely tricky because they originate deep inside the brain. You need invasive intracranial electrodes to record them properly.
Right, not something you can just do easily or non-invasively.
Not at all.
So how did they manage to look into this deep brain stuff in a way that felt, well, naturalistic?
This is where the study design was quite clever. They worked with 10 patients who had epilepsy.
Okay.
And these patients had already had these deep electrodes implanted as a part of their clinical treatment for seizures.
So it was an opportunity that arose from a clinical need.
Exactly. A unique window into the human brain's deep workings.
And what did they have these patients do while recording their brain activity?
It was surprisingly simple, actually. They just watched the first 50 minutes of the BBC series Sherlock.
Sherlock. Why Sherlock specifically?
Well, a narrative show like that has natural scene changes, right? Moments where the story shifts.
Yeah, like cuts between locations or plot points.
Precisely. And the thinking was that our brains naturally recognize these as event boundaries, like punctuation marks in the stream of experience.
Like a full stop at the end of a sentence.
Exactly like that. Signaling the brain, okay, close off that memory episode, let's start recording the next one.
Makes sense. And then how did they check if memories were actually formed?
After the viewing, they simply asked the participants to freely recall the plot. Just tell us what you remember happening.
And they compared that recall to the brain activity during the show.
Yes, and that's where things got really interesting. The patterns they found were quite dynamic.
Okay, tell me about these patterns. What did the ripple waves actually do?
Well, they saw different things happening in the hippocampus compared to those other cortical areas.
Right.
The hippocampus showed a definite increase in ripple activity right at those event boundaries, those scene changes.
So it kicks in specifically when there's a shift.
Exactly, which points to its role in segmentations. Like the hippocampus jumps in to say, okay, scene over, let's pack this up and consolidate it.
Packing and consolidating, got it. And the other areas, the temporal and frontal cortex.
Those showed higher ripple activity during the ongoing events themselves, while the scene was playing out.
So they're busy processing the information as it happens.
Right, they're dealing with the continuous flow of information within an event.
And then the hippocampus comes in at the end to wrap it up.
That seems to be the pattern.
That really makes the orchestra analogy click, doesn't it?
It does. The cortical regions are like the musicians playing the main theme, processing the ongoing melody.
And the hippocampus is the conductor, marking the end of one movement and the start of the next.
Beautifully put, ensuring everything is neatly divided and stored.
Okay, but does this actually link to remembering things better?
Ah, the crucial question. And yes, they found a significant link. Get this. Ripple occurrence in the temporal cortex, one of those processing areas, was significantly higher when encoding movie events that people later remembered.
Compared to the events they forgot.
Exactly. That strongly suggests the temporal cortex is directly involved in successfully encoding those memories so they stick.
That's a big finding. What about the hippocampus or the frontal cortex ripples? Did they predict recall too?
Interestingly, in this specific setup remembering movie plot points, they didn't find a significant difference for hippocampal or frontal cortex ripples predicting recall.
Huh. So the temporal cortex really stood out in this context.
It did seem particularly important for getting those specific narrative details locked in.
Did they look at how these areas influence each other? Like when one area ripples, what happens elsewhere?
They did explore that interaction too. It's quite sophisticated. When a ripple occurred in the hippocampus, the packer, there was a reduction in certain high-frequency activity in the temporal cortex.
Like it was quieting that area down momentarily.
Sort of, yes. And conversely, when a ripple happened in the temporal cortex, the hippocampus showed a decrease in a specific low-frequency rhythm called the alpha band.
And that alpha band is important.
Well, it's been linked in other studies to successful memory encoding, so it suggests this intricate back-and-forth communication, a real push and pull between the regions.
Wow. Okay, so stepping back, what does this all mean for, us?
Yeah.
For understanding memory problems?
Well, it has potentially profound implications. Traditionally, if someone has memory issues like amnesia, we often think about problems with attention or just difficulties learning new information in the first place.
Right, like they can't focus or the info doesn't get in.
But this research suggests another possibility. Maybe for some people, the problem isn't just getting the information in, but a failure in these segmentation signals.
You mean a breakdown in how the brain organizes and structures the information?
Exactly. A failure to properly punctuate the stream of experience so memories don't get filed away correctly, they just blur together.
That's a completely different way of looking at memory loss.
It is, and it opens up new avenues for thinking about therapies.
How so? What kind of therapies?
Well, imagine someone, perhaps an older person, starting to show memory decline, maybe presenting information to them in a more clearly structured way.
With deliberate pauses or breaks between key points.
Precisely. It might not just be about pacing. It could actually help their brain perform that segmentation process more effectively, giving it the clear punctuation marks it needs.
Directly facilitating the brain's natural encoding process.
That's the idea. It's a potential non-pharmacological way to support memory function.
Of course, research like this, especially with intracranial recordings, must have its challenges, right?
Oh, absolutely. It's crucial to acknowledge the limitations. These recordings are always guided by clinical needs first and foremost.
Meaning you can't just put electrodes wherever you want for research.
Correct. The location, the size of the electrodes, how long you can record, it's all constrained compared to what researchers might do in animal models.
And you mentioned anatomical differences earlier between human and rodent studies.
Yes, that's another point. Most human implants tend to be in the front part of the hippocampus, the anterior part. But a lot of the foundational rodent work on these ripples focused on a different area, the dorsal hippocampus.
So comparing findings isn't always straightforward.
It makes direct comparisons tricky, yes.
Is there any debate about whether these human ripples are the exact same things seen in mice?
There is an ongoing scientific discussion, yes. Are they identical to the classic sharp wave ripples in rodents, or do they overlap with other signals like high-frequency oscillations or something called theta-gamma coupling?
So the terminology and the precise nature are still being refined.
The researchers did carefully validate their detection methods, but they acknowledge this broader scientific conversation is still happening.
And one last thing, could they definitively say that more ripples at boundaries directly caused better memory in this study?
That specific causal link wasn't fully established here. Because ripples aren't super frequent in humans naturally, and you only get so many event boundaries in a 50-minute movie clip.
Not enough data points, maybe.
Essentially, yes. They couldn't definitively prove that the increase in boundary ripples directly predicted which specific events would be remembered later. More research is needed to really nail down that connection.
Got it. So wrapping this up, what's the big takeaway for someone listening?
I think the core insight is that our brains aren't just passive recorders. There's this active, ongoing process of segmentation, of editing, happening moment by moment.
Using these ripple waves as punctuation.
Exactly. And this active organization seems fundamental to forming lasting memories. It helps explain why some things stick so vividly and others just fade.
It really is a dynamic system, this orchestra, with the hippocampus conducting and the cortex playing the notes.
Absolutely. Different regions playing distinct but coordinated roles to create the coherent story of your life.
So this brings up a really interesting thought for you, the listener. If our brains naturally use, and maybe even thrive on, structure and these clear punctuation marks to form memories,
Yeah, what could you do with that knowledge?
right, could consciously applying this idea help your recall? Like maybe deliberately taking a short mental pause after an important meeting or learning something new.
Or even just structuring information you want to remember with clear breaks or sections. Could that help your brain do its packing and consolidating job better?
It's fascinating to consider. What stands out to you from this deep dive? Is there anything you might try applying, even in a small way, today?
Thanks for tuning in to the Health Headliner podcast by To Digital. Like and subscribe for more health news and breakthroughs. Stay healthy and informed. See you next time.